Yeah, I'm Sine. I'm from the Norwegian University of Life Sciences. I'm talking about evaluation of a SNP in the voltage-gated sodium channel as a possible pyrethroid resistance mechanism. So pyrethroids are believed to inhibit the voltage-gated sodium channel. From now, I will refer to it as NAV. In other arthropods, pyrethroids is commonly associated with a mutation in the NAV gene. This schematic overview, this schematic overview of the sodium channel shows known knockdown mutations in other arthropods described in the literature. The salmon louse harbor 3 isoforms of the NAV gene. No resistance-related SNPs have been found in 2 of them, whereas one resistance-related SNP has been found in the NAV13 previously. This SNP was found in 2 resistant strains from Scotland and not in the comparative sensitive populations. The substitution of isoleucine to valine in this position, marked with green, has previously been found in Drosophila melanogaster to decrease potency of deltametrin by electrophysiological measurements. To the left, the predicted binding site of deltametrin is shown. The isoleucine residue marked with the green circle corresponds to the SNP discussed in salmon lice. To the right, you can see a predicted model of the NAV13 protein with the 4 domains in orange, green, cyan, and yellow, respectively. The theoretical binding site of deltametrin, predicted from an in silico docking study, is shown. The SNP is marked with red there, and it's located close to the theoretical binding site and to other known knockdown mutations that are marked with purple. To further investigate the contribution of this SNP to resistance, 119 individuals from 14 different field and lab populations in Norway and from the Faroe Islands have been sequenced by Sanger sequencing. Identified genotypes of sequenced strains and their susceptibility to deltametrin are shown in the table. Multiple resistance strains have been screened, but only 3 sensitive populations. And the reason for this is that there are actually few populations that are not resistant to deltametrin. In general, the SNP was not found in the sensitive populations, whereas it was observed in some, but not all, of the resistant populations. Assuming an incomplete recessive mode of inheritance, where the heterozygous genotype confers resistance, this gives a classification success of 0.5. Thus, this SNP cannot be the sole cause of pyrethroid resistance, although some contribution cannot be ruled out. Pyrethroid resistance in the salmon louse has previously been associated with, pyrethroid resistance in the salmon louse is maternally inherited, and several non-synonymous SNPs in the mitochondrial DNA has previously been associated with resistance. Follow-up studies are therefore in progress to screen for SNPs in NAV13 together with mitochondrial DNA. And this will be further discussed by my colleague Linas. Electrophysiological testing will also be performed on the wild type and the mutated variants of NAV13 to look for differences in sensitivity. We are also working on identifying alternative splice variants and potential other resistance markers in the NAV isoforms by a novel method including 3rd-generation sequencing. that Nadia, my colleague, will talk more about now. So thank you for your attention.